Good students. This is Wilson. First of all, welcome back to our YouTube channel, Physics for Backbenchers. Okay, in today's class, we are going to study cell cycle and cell division. Remember, students, it is one of the most important chapter for NEET examination. And from this chapter, we can predict minimum 1 to 2 questions. Okay, so already we discussed about cell and its cell, the basic unit of life. And in that chapter, we discussed about different kinds of cellular organelles and including nucleus. So now let us see how the cell is going to divide and what kind of mechanism is present in this cell division. So first of all, let us see, we are going to study an introduction. First, this mitotic division was studied by Walter Fleming in salamander. Okay, cell division include both mitosis and meiosis. First time, this mitotic division in salamander was studied by Walter Fleming. And one more scientist, Strasburger, he discovered meiosis. Okay, and also he gave the names like prophase and metaphase, anaphase, and telophase. Okay, I hope that you are understanding. Mitotic division first studied in salamander by Walter Fleming, and Strasburger discovered meiosis. And he also given the names, which names are like prophase, metaphase, anaphase, telophase, which are the subphases of mitosis. And these name meiosis was given by the scientist Farmer and Moore. Okay, Strasburger discovered meiosis, but its name was given by Farmer and Moore. So now let us see what kind of cell division types are there. So basically, these cell division is divided into 3 types in all kind of organisms, and these are amitosis and mitosis and meiosis. So in this cell cycle and cell division, step by step we are going to study here. First, we are going to study about this amitosis process, and then followed by mitosis and meiosis. Remember, it is one of the most important and little bit confusion. That is the reason strong basics is important. Okay. So first of all, we are going to study about amitosis process. This amitosis process was described by Remark in the year of 1841. He discovered this amitosis process. So when compared to mitosis and meiosis, it is a very simplest mode of cell division. And but in which kind of organisms can be see these amitosis? Of course, it can be seen in lower organisms, like especially in prokaryotic organisms. So these amitosis process starts with the karyokinesis, and it ends with the cytokinesis. So karyokinesis means nuclear division, and whereas cytokinesis means cytoplasmic division. Okay, so remember once again here, it is simplest mode of cell division compared to mitosis and meiosis, and this especially these amitosis process starts with karyokinesis and it ends with the cytokinesis. So let us see what are the changes which takes place especially inside the cell. So initially, what is going to happen here? Elongation of nucleus occurs inside the nucleus. Suppose just you can imagine this is a cell and this is a nucleus, and whereas nucleolus is present. So initially what is going to happen here? So first of all, the first change can be seen in the nucleus. So initially elongation of nucleus can be seen. So in this shape, how the nucleus is look like? It is look like dumbbell shape. Okay, and afterward these nucleus gets divided into 2 daughter nuclei. Okay, so with this karyokinesis part is completed. It's very simple, simplest mode of cell division, and where initially elongation of nucleus takes place. As a result, it is going to convert as a dumbbell shape. As a result, formation of 2 daughter nuclei. So with this, karyokinesis is completed, and it is followed by cytokinesis. Okay, So this nuclear division is followed by division of cytoplasm. So as a result, the formation of two daughter cells takes place. So right now, this cytoplasmic formation takes place. As a result, two equal halves of daughter cells are formed. So these daughter cells are exactly identical to the parental cell. Okay. So and especially note here, there is no spindle fiber formation here and no distinct chromosome formation. Okay, so in examination point of view, sometimes they will ask you questions like this. In A mitosis process, what kind of changes cannot be seen? As obviously, there is no spindle fiber formation and followed by no distinct chromosome formation. So these daughter cells are approximately equal halves of a parental cell. So let us see in what kind of organism this mitosis process takes place. Mitosis process takes place in BGA. BGA stands for blue green algae, and followed by PPLO. PPLO stands for pneumonia-like organism, and next one is bacteria and other eukaryotic cells. Okay, and one more question is very important. Suppose in yeast, suppose these yeast cells, they reproduce asexually by budding mechanism, and it is a type of amitosis. Okay, so in yeast, budding occurs by a mechanism called amitosis, and in especially amoeba, in an amoeba, multiple fission occurs by amitosis process. Remember, in yeast cell, budding occurs by amitosis, and whereas in amoeba, multiple fission occurs by amitosis. So similarly, like suppose if you take mitochondria and chloroplast, so these also divide by amitosis process. Not only this one, and but also if you take in mammals, growth of fetal membranes like amnion, chorion, yolk sac, etc. So these all they divided by amitosis process. So let us see what kind of questions you can predict from this amitosis process. First of all, it is the simplest mode of cell division, whereas it starts with karyokinesis and ends with cytokinesis. So which occurs in especially prokaryotic organisms and followed by eukaryotic cells, and in yeast cell it occurs by budding occurs by amitosis. In amoeba, multiple fission takes place by amitosis process. Okay, so just you need to focus on examples and the mode of the mechanism, the way how they are going to reproduce. Okay, so with this amitosis process is complete. Okay. And another mode of cell division is mitosis, and so in animal cells, this mitosis was discovered by Walter Fleming. Okay, in animal cells, the mitosis was discovered by Walter Fleming in the year of 1879. And whereas in plant cells, this mitosis was discovered by Strasburger. Remember, students, Strasburger also discovered meiosis process, and the name and also he given the names like prophase, metaphase, anaphase, telophase. So in animal cells, Walter Fleming discovered mitosis, and whereas in plant cells, Strasburger discovered meiosis process. Okay, and how can you define these mitosis? So mitosis is a type of cell division in which parental cell, suppose just you can imagine this is the parental cell, which divide to form two daughter cells. Okay, mitosis is a cell division in which parental cell divide to form two daughter cells. This is called mitosis. Do you think it is very simple? Of course, there are many sub pages are there, and there is a mechanism in this mitosis process. So that one I'm going to study. So this is the definition. And in what kind of cells these mitosis process can be seen? Of course, it can be seen in somatic cells. It occurs in somatic cells. Somatic cells like, which occurs in haploid cells and also diploid cells and polyploid cells. Okay. And these mitosis process also called as indirect division. Why it is called indirect division? Because it occurs in somatic cells. It do not takes place in germ cells. Okay, so that is a major reason, and it is very important to increase the number of cells in our body. Okay, so that is the reason these mitosis which promote our body growth. So why we are calling it is indirect division? First of all, meiosis takes place, and these meiosis which is followed by mitosis. So in meiotic division also called as a direct type of division. So whereas these meiosis also called as the indirect division. So let us discuss right now cell cycle mechanism. It can be divided into two phases. Okay, in the mitos total cell cycle can be divided into two phases. They are number one, interphase, and followed by M phase. M phase stands for mitotic phase. Okay, so first of all, we are going to study about interphase, and then followed by M phase. M phase stands for mitotic phase. So remember here, first of all, what is mean by interphase? Okay, so interphase which includes G1 phase and followed by S phase and G2 phase. So in the given picture, G1 phase and S phase and G2 phase. So these all pages are included. So it is divided into three substages like G1 phase where G stands for gap, gap 1 phase, and S stands for synthetic phase, and G2 phase. Okay, and the way how you are going to remember these G1 phase and S phase and G2 phase, so you can able to easily solve the questions. You can able to easily answer the questions in NEET examination. Okay, and this interphase also called as the longest phase of cell cycle. Already I told you the entire cell cycle is divided into two major phases. They are interphase and M phase. Right now we are studying in an interphase. Okay, and interphase is the longest phase of cell cycle. Approximately the duration is 19 to 22 hours. Remember, students, where I have taken human cell as a reference. Okay, and human cell division which takes place 24 hours approximately. So among 24 hours, see 19 to 22 hours it will take place. Okay, and it is also known as a resting phase. Why? Because actual division which takes place in M phase, what do you call in mitotic phase, that is the reason it is a resting phase. And during this phase, so during an interphase, cell is metabolically extremely active. Okay, so metabolically extremely active. Why? Because where one cell is going to divide into two daughter cells, that is the reason it is hyperactive. Okay, there is a lot of difference between active and hyperactive. So during this an interphase, the cell is metabolically extremely active. So now let us discuss what is mean by quiescent phase, or we can say G0 phase. Generally, of course, already the cell cycle which divides into an interphase and M phase. Suppose this is a cell which is divided into two daughter cells, and now one more stage is there. Means after completion of these M phase, immediately this cell which enters into G1 phase. But in this stage, it is going to arrest further. There is no division. That is the reason it is also called as a G0 phase. Suppose I need only two daughter cells. Afterwards, I don't need any further division. So in such conditions, the cell will not be dividing further. So in such conditions, which is also called as quiescent phase or G0 phase. So G naught phase, also known as inactive stage of cell cycle, where it is very important. Cell is metabolically active, but do not divide or do not proliferate. Okay. So quiescent phase generally can be found in permanent tissues. Generally, in these permanent tissues, further cell division cannot be seen. Okay. So this is called quiescent phase. So when you can able to see this quiescent phase after completion of mitotic phase. Okay. Already in previous examinations also we can able to see some of the questions on quiescent phase. That is the reason to focus carefully. And what is the duration of a human cell division? I told you already 24 hours. Generally, these cell division, which is going to vary from one organism to another organism. Generally, in case of the human beings, the human cell, to convert one cell into two daughter cells, it will take 24 hours. Suppose this is a cell, it is a parental cell, and whereas which is converting into two daughter cells, so the duration is 24 hours. Suppose if you take yeast, it is a fungi, it is a unicellular fungi, which takes place 90 minutes. Within 90 minutes, one parental cell, which is converted into two daughter cells. And bacterial cells approximately 20 minutes. Remember, it is the fastest cell division. Fastest cell division can be seen in bacterial cell. Okay, fastest type of division. So that is the reason cell division, which is going to vary from one organism to another organism. In human beings, cell division takes place 24 hours, and whereas in yeast cell 90 minutes, and in bacterial cells approximately 4 to 20 minutes. Okay, and next one is cell cycle running by a special group of proteins called cyclins and cyclin dependent kinases. CDKS stands for cyclin dependent kinases. Okay, suppose if you take here this is a parental cell, and now this cell alone itself, which is not dividing. Some proteins are there, they initiate, they take responsibility to divide. So what are there? These are cyclins and cyclin dependent kinases, which also called as MPF. MPF stands for mitotic promoting factors. Remember, so in examination point of view, if they will ask you questions, so generally, which kind of proteins they initiate cell division means cyclins and cyclin dependent kinases. Remember, so these proteins these are the special proteins which are present inside the cell. If these proteins are absent, the cells cannot divide. Okay, so with this introduction about interface is over. Now let us discuss one by one the sub phases of interface. So that already I told you there are three sub phases in the interface. First of all, G1 phase, and followed by S phase and G2 phase. So in an examination point of view, this sequence also very important for you. What is the sequence? First of all, G1, and then followed by S, and then followed by J2, then followed by M. Okay, so this is the sequence. There are many questions were framed based on this concept. That is the reason this is the pathway. First phase is in the interface G1, and followed by S phase, and followed by G2 phase. Then after which enters into mitotic phase. Generally, this mitotic phase, the duration is approximately 1 hour. Okay. So let us see here. First of all, these G1 phase. So it is the first phase of an interface. Okay, it is the first phase of an interface. Just I will tell you only one word about these G1 phase. Remember what kind of changes can be seen in G1 phase. Remember, cell growth occurs. Cell growth occurs. Okay, and it is the duration of this G1 phase is approximately 16 to 15 hours, and it is most variable phase. Okay, and remember, it is the longest phase of interface. So I told you when you take the sequence here from G1 to G1S and G2 phase, so this G1 phase is the longest phase. Okay, it is the first phase of an interface, and it is the longest phase, and it is the most variable phase. Okay, and generally what kind of changes can be seen inside the cell here? Yes, cell is metabolically highly active here, and cell growth occurs. Okay, next cell organelles like mitochondria, in case of the plant cell, chloroplast, Golgi apparatus, endoplasmic reticulum, all kinds of organelles are synthesized. Okay, cell organelles are produced or synthesized during S phase. So remember and do not get any confusion. And next one is whatever the structural and the functional proteins, okay, are formed. Okay, and already I told you nucleolus is present. These nucleolus which produces ribosomal RNA and mRNA and tRNA. And next one is a cell is metabolically active, and which is also called as pre-DNA synthetic phase, which is also called as a pre-DNA synthetic phase. So in the given picture, this is a G1 phase. So all kinds of activities which takes place here. Okay, protein synthesis takes place, and organelles are produced, and increase the volume of cytoplasm. Increase the volume of cytoplasm. Okay, so why? Because suppose this is a parental cell, and whenever cell division occurs, two daughter cells are equal to the parent. That means what is going to happen here? The cell size, the volume of cytoplasm is increased. Okay. So in G1 phase, remember what are the key points here? First of all, cell growth occurs, and cell is metabolically active, and nucleolus produces like ribosomal RNA and mRNA and tRNA, and it's the most variable phase and the first phase of an interface. Okay, and next one is S phase. S phase stands for synthetic phase. So in which phase especially DNA replication takes place. Okay, generally most probably they will ask you, this is an S phase. So what is going to happen here? DNA replication takes place. Okay, so in this phase the amount of DNA doubles, but chromosomal number but remains same. Suppose if we take, there are 23 pairs are there, where the DNA becomes doubles, but the chromosomal number is remaining constant. It is very important. First of all, do remember in S phase, what happens? DNA replication takes place, and chromosomal number as such, and the duration is 6 to 10 hours. Remember, already I told you G1 phase is the longest phase, but in case of the human beings, longest phase, S phase is the longest phase. So this is an exception. And at the same time, even histone protein synthesis also, histone proteins also synthesized in S phase. And next one is the centrosome. It plays a vital role in cell division, including centrioles, replicate in late S phase. So these are the 2 points. First of all, in an S phase, first what is going to happen here? DNA replication takes place, and it is the longest phase in case of the human interface, and centrosome duplication and histone protein synthesis takes place. Now, how to remember, how to understand both G1 phase and this S phase? So here I told you some proteins are there. Which proteins? Structural and functional proteins are formed or synthesized. And I told you here, it is this G1 phase also called as pre-DNA synthetic phase. Suppose if you wanna prepare any chicken, suppose you are going to prepare chicken curry, so you require some ingredients like chicken is required, and oil is required, and some other ingredients like vegetables are required. That means we are going to prepare something. So here the major important task is DNA replication. To do this DNA replication, whatever the required enzymes are there, like RNA and other enzymes, and everything are produced in this G1 phase. So like that, you need to remember. So what is going to happen in G1 phase, and what kind of changes can be takes place in S phase. Okay. And next followed by the last phase of an interphase is a G2 phase. So it is the duration is 3 to 12 hours, and it is also called as post DNA synthetic phase, and where tubulin proteins are synthesized. Okay, and which starts formation of spindle fibers, and cell division. Supposed to drag one chromosome to the opposite pole, 24 ATPs are required. That is the reason an abundant ATP is going to store in this G2 phase. Okay, so remember, first of all, everything takes place, all kind of activities takes place in G1 phase and G2 phase, and followed by the post DNA synthetic phase, and some proteins are synthesized in this G2 phase, and followed by which is going to store ATP molecules, and sometimes some whatever the cellular organs required, again they are synthesized, and everything. See, this is the last, the post DNA synthetic phase. Now it is going to enter into M phase. That is the reason all kind of checking points are there. Checking point means what you call here, there are whatever the errors are. Suppose in DNA in S phase the DNA replication takes place, whether the replication takes occurs properly or not, everything is which is going to check. Okay, twice the errors can be what do you call evaluated in this G2 phase. Some potentials are there, they are going to evaluate. If everything is perfect, suppose if everything is perfect, then which is going to enter into the actual cell type of cell division, which is called mitotic phase, or we can say division phase. Okay, so this is called about an interface, and where three kind of subphases are there. These are the G1 phase, and followed by S phase and G2 phase. Okay, so just do focus on first of all, I will tell you only 3 points. In G1 phase, cell volume can be increased, and followed by in S phase, DNA replication takes place, and centrosomes they replicate, and histone proteins also synthesized. And in G2 phase, further some whatever the enzymes are required for mitotic, they can be synthesized and properly, which is going to check. Why? Because an important task which occurs in a major task can be takes place in this mitotic division. That is the reason everything, each and everything they are going to check. So they can rectify each and every mistake. Okay, if everything is perfect, suppose if G1 phase is perfect, then which enters into GS phase. If S phase is perfect, then which enters into G2 phase. Suppose there is a drawback once again in this either G1 or S phase, and this G2 phase further cannot allow the cell into M phase. Okay, so that is the reason. If everything is perfect, then only the G2 phase which enters into an M phase. Okay, so this is about and this interface, and in tomorrow's class we'll be going to study about mitotic division. In the mitotic division, I'll explain you about prophase, metaphase, anaphase, and telophase. So thank you.